wouldn't be talking much about, yeah, I wouldn't be talking much about Dr. Ash because Dr. Bala and Adrian may be able to, because they have been in person with him, so share things about him. But going through his profile, it's really amazing. He started as a student of physics. I think that has given a lot of impetus to his innovative mind and his modifications of things and procedures in the future. So that I found in his biodata. Then also he has been under Dr. Willem Koff, and I used to be um, impressed by Dr. Willem Koff again because of his great work in interventions and trying to bring in a lot of across specialities. And so you see here, he has BA Physics, then he goes on to finish his MD, then postdoctoral fellow in physiology in Indiana University, then fellowship in nephrology. And his fascination with renal replacement therapy, made him enter into each facets of dialysis therapy, right from access, right from hemodialysis to peritoneal dialysis. And the last one which I came across during the ASDN meeting this time was a PD catheter which I had designed, which has not hit the market yet, and I'm sure he would be describing it. And he has his own company where he is the chairman, uh, a board of Ash Access, it's called, and he's a co-founder of numerous spin-off companies from there, and is again the director of dialysis program in IU Health Night Lafayette, and where he was responsible for assuring quality care, and he was, I think, one of the co-founders of one of the person who started ASDIN in US long ago. So we are very happy that you are here, sir. And I've been wanting, and specifically me, I very proudly have a photograph I show in most of my talks, and even in my room I have the prize I received during one of the ASIN meetings for its time for, I think, AV fistula, or its time nephrologists enter cath lab. So I won that paper, won a prize, and he was the one who gave me the prize, and I proudly store that photograph. I think uh, Dr. Bala and Adrian, you have anything to say about Ash before he starts?
Maybe I'll just sorry, Adrian, go ahead.
I just think he's the epitome of humility. He has a lot of wisdom to pass on. I always tell him, every time I see him, he reminds me of that actor, Charlton Heston. I don't know, I've told you this many times probably, but it's your humility, I think, that I take the most from you. I know you have a lot of wisdom, you have a lot of experience, and you're just an all-round nice person. That's what I wanted to add. Besides all his accomplishments, I wanted to show the human side to Dr. Ash. He can sing, he's very smart, and he's very humble.
My 2 cents is, first of all, I wasn't even born when he finished his fellowship. So, neither do I have the age or the wisdom to, comment on Dr. Ash's accomplishments. In addition to what Adrian said, it's a burning passion on innovation, which is truly commendable. They have accomplished so much. Even in the last conference, Dr. Hash had a new concept on what he wanted to put forward from an innovation perspective. Just maintaining that passion towards innovation in just one field for over 4 decades is just mind-blowing to me. I hope all of us take that inspiration and maintain that passion towards innovation. And when you're innovating, maybe like 20% of the times or less than that, you will cross the finish line. But the rest of the times you're just, trying to get there, and you probably will never get there. And Dr. Ash maintains that perseverance, and that's truly inspiring to me. So that's my 2 cents. Thank you.
Well, thank you. And to that, I would mention that there is a fine line, however, between dedication and obsession, and sometimes you could cross that line without knowing it. But thank you. The road to invention is loaded mostly with new learning and discovery for yourself. I mean, you learn an amazing amount trying to put these things together. Great. Well, I thank you so much for the invitation, both Dr. Ballas, and really appreciate the chance to talk.
And before you start, I mean, now that people have joined, see, Dr. Georgie has joined us for a long time, and I thought it was very right to have him as a faculty today, because he was the one who started peritoneal dialysis in our country, and there is going to be a lot of mentioning about PD catheter insertion and the innovations what Dr. Ash has done in this talk. So thank you, sir, for joining this meeting.
Thank you, Bala, for the kind invite.
Right. Yes, yeah. Dr. Ash, I think you can start your presentation.
Oh, okay. Well, thank you for the invitation. And it was so broad, you wanted me to cover most everything I've done, and I really didn't want to bore us for over amount of time. But I will give you a few lessons from what I really have learned. So you've already gone through most of the history. I would say I did start out my practice in nephrology and my research experience about the same time in 1975 when I came to Lafayette. And I stayed with the same multi-specialty practice group as it expanded for 47 years. I just required it a couple of years ago. But also to keep my efforts going in research, I worked first with a bioengineering center at Purdue, developing mostly new approaches to hemodialysis. And then I met this guy, Bob Truitt, in 1983, who decided with me to form a company to pursue this research. And that's changed just about everything. What was Ash Medical became HemoCleanse, became that and Ash Access. And we ended up basically creating several sister companies, which ended up finally with a license of the Renal Solutions Company's product and the sale of that company to Fresenius and the sale of a spinoff related to the potassium sorbent. ZS Pharma was sold to AstraZeneca. So what caused me to pursue these inventions and projects for so long? Well, everybody knows that necessity is the mother of invention, but who is the father? I often ask that. And I can tell you from experience, frustration is the father. Personal frustration about the 10th time that a medical treatment fails to be effective and causes results worse than the disease you are treating. You say, somebody's got to fix this. And you literally can't stand it. So you look around, if nobody's there, you take off. So when I looked around dialysis in 1975, there were all kinds of problems with successful therapy, comfortable therapy, and especially long quality of life. So I decided to look at two of them in particular. One was blood access devices. Especially needles bothered me, but catheters also had problems. And also the huge volume of water that we use in dialysis, over 100 liters of purified water for standard treatment. Those seemed things that I thought I could fix. Just a real short history, because I'm actually to speak at the call celebration, a few weeks in Kempen of his 80th year of after the successful recovery of the first dialysis patient with acute renal failure. So anyway, I threw in a few of these to remind us that when he started, we had the rotating drum kidney, cellophane tubes that would expand and close and propel themselves, small units of blood through the machine. And his big advantages came from materials that he found that other people hadn't really had a chance to use. Cellulose, which was used in sausage casing, and also heparin, which was finally a reasonable and fast, effective anticoagulant. But other improvements in the machine that really made it work at all really came from the Kolff and John Merrill, George Thorne. He collaborated with them, and they're the ones that really made it a full machine with a blood pump, pressure actuated, easier exchange of dialysate, better warming, etc., to at least make this complicated procedure life supporting. And finally, that was evolving in the 50s and early 60s to finally disposable dialyzers with cellophane compressed between plastic screens, but something you could buy, use once, and then throw away. But really, a more clever and ingenious solution was the plate dialyzer, which came from Alwal in Sweden, which had flat sheets of membranes. And because this membrane supports were so finely designed, they could allow a huge amount of surface area, like a square meter, to be held in position that it would cover 300 milliliters of blood. And they had low resistance. So this was the foundation of home hemodialysis, finally, both in United States and in Netherlands. And this is about when I came in. This is the first dialysis machine I ever saw when I was a med student at the University of Kansas. And this was the Travenol recirculating machine. It was really the first integral machine. Cleverly, it had huge amounts of salt water dialysate in the lower tank, but a fat rapid flow of a smaller volume through the dialyzer that was heated and then discharged. Eventually, of course, this got replaced by the current hollow fiber dialyzers with all kinds of advances. People sometimes say that there weren't great advances after 1960. Well, that's really not true for dialysis. I mean, if you look at the advent of controlled filtration, minimal blood volume, bicarbonate dialysate, for example, those things were really vital to making it something that you could bring in center and successfully treat patients for 3 or 4 hours, 3 times a week. Now we're learning that that's probably not the best of schedules. Certainly more time and slower would be better. But anyway, so here's where I came in, plus the Ready machine. The Ready machine was, to me, truly remarkable because I'd done enough research in sorbents as a med student and fellow to realize that there are thousands of toxins of uremia. And to think that a simple column basically could be used to regenerate 6 liters of tap water during dialysis and remove all of the significant uremic toxins, at least the ones we could measure then, was just truly amazing. And it really did it by very simple components. Excuse me. Activated carbon for all the organics, the thousands of things. Urease layer to convert urea to bicarb and ammonium. A cation exchanger that was loaded with hydrogen and sodium in exchange for that. And then an anion exchanger to pick up phosphate. We're now learning there's a whole lot of other organic anions that are very important uremic toxins like oxalate. So it probably worked for that too. In fact, Dr. Kolff that I worked with once told me if he had known there were so many uremic toxins, he never would have invented dialysis. He said, I would have just given up. But the non-specificity of these components is probably really what bailed us all out and is still bailing us out. So anyway, I was hooked on sorbents by that time. So in 40 years of R&D after the companies were created, our company basically created 12 new devices and drugs to make hemodialysis better. That is simpler, safer, easier to use to do at home. So I was eventually kind of ticked off at the outcome of some of these projects because surprising to me then with each project, basically most of them did not end up changing the practice of medicine with some exceptions. So of course I wrote a book. I'm convinced by this time that most people who write a book are really just ticked off about something. And in fact, really, if you're really ticked off, you write 12 editorials for artificial organs in 12 months, one about each topic. So that's what I did. I wrote the editorials. They were published in Artificial Organs in 23, and then I put them together in the book, plus a bunch more. So anyway, without going into huge amounts of detail, so here's the 12 projects, from wearable artificial kidneys to carbon block and sorbents that are still going on now. And here's the stages of development. You don't think about it often, but there's a lot of stages to go through in going from conception to modeling, prototype, patenting, and lab testing. All of these projects sailed right through all that stuff. The surprising thing was, and by the way, the blue ones are the sorbent-based dialysis systems, and the red are the intravenous catheters and related stuff. The surprising thing to me, so far so good, was the number of projects that got clear that far, clear through animal trials, FDA approval, and then still didn't make it to changing the practice around me. And my measure of success was not me, but when my partners started using the procedure, that was rewarding, or the drug. So if you look at the causes of failures, only one really failed in animal trials, possibly two if I look at it another way. But there's government regulations. There's redesign of the product after it's licensed by a major company. There's a real huge risk point. Then there's marketing. Is the marketing really logical? Are they going after areas they can easily sell the physician, are scientifically supported, etc. And even some of them, some have failed to be successful in the U.S. when they're very successful around the world, which is really weird. So two projects, however, were dramatic successes. One was the zirconium cyclosilicate, the world's first monovalent selective cation exchange crystal that is now out as Lokelma. And then the second, of course, was the split cath, which amazed me that it became so popular. So here's a real brief summary. I actually spent my first 3 months as a real nephrologist in a real job at the University of Utah with Dr. Kolf, because I had to go out there to see what happened to the wearable artificial kidney. And it was a great experience. His team was amazing. And I'd really never seen engineering research. I mean, I grew up in the scientific biochemical area, but to see a team all dedicated to different parts of a machine and see it come together, that was pretty cool. Anyway, his was truly portable, but only if you had charcoal in the dialysate. When you added, to get rid of all the other things, the simple small molecules like urea, phosphate, potassium, you still needed a dialysate. But it was successful clinically, but when he licensed it, the company tried to pound it back into a regular in-center dialysis machine. It just didn't work. So when I set up at Purdue, I pursued my own sort of reciprocating dialyzer called the SSRD, which was really unique. In fact, mostly because it borrowed the same physical effects of what happened in the old rotating drum kidney. The membranes would expand as the blood went in and then collapse. When it collapsed, it was lousy for transfer, especially of middle-sized molecules. But when they open, they get fresh dialysate right at the membrane surface, getting rid of all the stationary layers. So anyway, that thing was flowing really smoothly until animal trials. And there was something in dialysate that we couldn't find in the lab that would solubilize the aluminum silicate from the project. That was gone. So we went with a more conventional ready column, basically, and that became the Biologic HD. We threw into this 1983 project everything we could think of that would make dialysis better. Sorbent-based, single access, plate dialyzer as a blood pump, so there wasn't even a blood pump. An airless blood circuit, controlled filtration, automatic fluid replacement, and it was a perfect nighttime home machine. What happened? The same year we got FDA approval to sell this and had 40 machines in progress. CMS decided to eliminate home dialysis helpers. There were 4,000 hemodialysis patients in the United States, and in a month or two, there were 400. They all went back in center. So our venture capital companies walked and said, well, do the other thing you were talking about, excuse me, which was an artificial liver. So if we took the column out and we put in a suspension of charcoal and cation exchangers, we felt that we could treat liver failure. And in fact, it really did work, especially for acute on chronic liver disease and acetaminophen overdose. So we started a company to pursue this, couldn't find a marketing group otherwise. And when they originally had a plan to market that device, it was going to be focused on 5 liver transplant centers in the United States. They were already working with us in research. But as soon as they got venture capital money, they decided to expand and shotgun the market. So all of a sudden, there were 50 centers that were using this machine and not using it in the right kind of patients chosen logically. So the hemotherapies went bankrupt, and I stewed around for a while. And then remembered Stange and Mitzner that had visited my lab a few years earlier, checked up on them, and they were building their own artificial liver called Mars, which I look at as a sorbent-based system, kind of a recreation, but an improvement in what we had. So like a lot of things, the way to be successful is to redefine your definition of success. And I define it as if somebody steals it or borrows it and makes it a success, that's mine too. So in around 2000, we decided to resurrect the old HD, biologic HD, in fact, with a hollow fiber dialyzer, but still having pressure actuated blood flow. So we had a little tiny miniature artificial heart thing to propel it. Under controlled pressure, you don't need pressure gauges, you just monitor the flow. And controlled filtration, automatic fluid replacement, that was all there. Renal Solutions was sold, as I mentioned, to FMC, and they just had to fulfill their dream of a sorbent-based system for everything, including in-center 3 to 4 hour dialysis, which was really impractical from size of sorbents, chemical changes. So eventually, in that redesign, they canceled the project. So we finally come to these really successful one. This developed out of 40 years of collaboration between our lab and Union Carbide. And the deal was we had failure with the first sorbent system, that SSRD, and I made a deal with John Sherman, their director of research, that if they go back to the lab and they can make a monovalent cation selective cation exchanger, I'll test it in animals for free. So he called me one day around 2000 and said, I got it. And I said, you got what? He said, what you told me, you need a monovalent selective cation exchanger. And sure enough, he sent it. We tried it in 5 rats because that was all we could afford in our little facility. And it was just astounding, the amount of potassium and ammonium that it removed, affecting the urea levels in the urine. So anyway, we stayed close to that ZS spinoff we formed, at least to get them through the first clinical trial. And then they were kind of on their own. Eventually, they were sold. It's now Localma. And then, of course, there's a split cath. I've often remarked that I spent 15 years developing an artificial liver device, and nobody could care less, it seemed like. I spent 15 minutes on this thing, put it in an envelope, mark the envelope to Tony, their president, make this, Steve, and there became the split cath. So you never really know. Well, anyway, it ended up being even more successful than I thought. But we did have a split with MedComp, which was unfortunate because we apparently didn't define well enough in the license what's a kit and what's not a kit. When you charge for the catheter, it might be a $30 item or $50. And charge for the kit, it's like $200. So it's a big difference. So anyway, that we still have good relations now. But the next catheter I developed, the Centros catheter, with help of Dr. Bala, by the way, then went to Merit instead of. So here's a side short track, because I promised Dr. Bala that I would talk a bit about vascular access, just for interest. If you look at really the genesis of the different types of catheters we have, it's partly based on the logic of what the catheter should look like in size, but it's also based on how you're going to put it in. And acute catheters were really designed to go in by a Seldinger technique over a needle and a dilator. And that's the reason that they're usually fairly stiff, and they're pointed, and they don't have a cuff because they're acute. So there with the Seldinger technique, that's how I learned dialysis as a resident and fellow, was you put 2 of these things in every time you dialyze somebody and just come out at the end of treatment. You put them in again the next day. You got really good at it pretty quick. So the first single 2-lumen catheter was the Udall catheter. And I actually used that as well. And it was a concentric thing. So you had 1 lumen for return in the middle and 1 lumen for drawing blood outside. Very clever, but unfortunately not the best designed hydraulically. So the resistances were pretty high or the flows were pretty low. It came along Maherkar, whose paper in ASIR journal was astounding to me. On a theoretical basis, he showed that the double D catheter with the flat wall in the middle is hydraulically much more efficient than any other design you could have for the same size catheter. And so that's become really the standard. That was the standard for split cath. All I did was a pretty obvious thing of trying to make one catheter end into 2. And then the Centros was 1 catheter end into 2 and bent to the middle so that the tip didn't contact the wall and suck against it. So those were the acute catheters. The real genesis of these soft, blunt, cuffed catheters, chronic catheters we call them, was the idea of using a split sheath rather than the straight Seldinger technique. And I don't really know where the split sheath came from, but some engineer deserves congratulations somewhere for figuring out you could split Teflon down the sides just by pulling it in a tube. But with that in ultrasound came the Canode dual catheters. But the market needed a single catheter, hopefully. Quentin came out with the first one. It was an oval, about 20 French total size, too big. Maherkar made it a DD shape. It was more practical, and that's really still the step tip catheter today. The Ash split cath and Ash Centros have both come out in chronic form immediately. And of course, the Tal palindrome is really quite clever, I think. I should mention that I'm still looking for the perfect long-term access that won't have any effect from sheathing. And believe it or not, this little gizmo, it's right here somewhere. Sorry. Well, it's in another bag. Anyway, oh, here it is. So this little thing is actually a catheter which has a lumen for drawing blood that's just right at the surface of the interior part of the vein. It's not touching anything. It's not, there's no catheter contact with the wall on the other side, so you can't get a sheath around it. But it's just hanging out there in the middle of the flowing blood. And the return catheter, this short little limb on the bottom, will just rest and direct the return blood downstream. It's still a research project, but the animal trials of the single version of the single lumen version were really, I thought, quite impressive. And where have we gotten with chronic arterial access? Well, some remarkable designs. Quentin Scribner's shunt, the fistula, basically the graft was I think a tremendous advance. And of course, the Hero is certainly interesting. I knew it had a place in the market when I first saw it, but it wouldn't be the standard, hopefully. But what's interesting is what's next, and that's one thing that where we can, there's still, as a partner of mine once said, there's plenty of room for invention here. So getting back, so related to the catheter, of course, I was really concerned about the other big problem besides flow, which was infection. So around 2000, we discovered that actually if you use a little more concentrated citrate, such as 23%, it's actually very bactericidal. So the same exact compound could be used to keep your catheter open as to keep it free of infection. And this is an interesting story. I'll try to make it short, but basically we got two patents, one in the US and one in Europe. And the patent was contested by the company that was a licensee for this product of concentrated citrate. And on appeal, they won. And the judge said, "You're right. This patent on citrate is invalid because citrate's been around, since the sailors' days." So anyway, in the US, there was one physician placing a catheter who made a horrific overdose of the citrate against all the recommendations and logic. And as a result, it's still banned in the US as a catheter lock material, but popular elsewhere. So that shows you're never in the end zone until you're in the end zone, right? So we didn't give up on catheter locks. The next big approach was Zergen. I said, okay, we'll use 7% concentrate, citrate, and add a couple of antiseptics compounds, and that will be effective. But it was way more effective than we thought. In fact, tremendous synergy between all three components. So we did a huge randomized trial, and the results were easily publishable and impressive in diminishing infection. Like, truthfully, most antiseptic catheter locks will drop infection rates 3/4. But the FDA always has a drug reviews by the device division and always has a device reviewed by the drug division. And the drug division decided this is a drug. And as a second drug, an unwritten rule of the FDA is you have to have 2 clinical trials, not 1. Well, the licensee decided they didn't want to afford a second clinical trial, and therefore they abandoned the project. So we then adapted it to use as a skin prep, which was more effective than chlorhexidine. And that's still out there. We haven't quite decided how to license it yet. But more recently, believe it or not, we did a third trial. We're now very interested in a drug that just has sodium citrate and benzyl alcohol, which is in these little vials. It was on the market actually in the US, but the COVID epidemic or endemic now led the company that was a compounding pharmacy into filling up all kinds of drug shortages, so they never made a second batch. So currently it's not, but there is significant interest in this in the Pacific Rim, particularly the Philippines and some in India, to bring it back. It'd be very inexpensive and hopefully have kind of the same market price as heparin, but be more antibacterial. So finally, we get up to the 12th project, which is actually still ongoing. I've always felt that if you could just find 5 toxins in the gut, 5 chemicals, which would be urea, phosphate, potassium, sodium, hydrogen, then everything else that hemodialysis does chemically could be taken care of by a carbon block column, which would be a very simple thing to add to your dialysis system. So all you need is a dialyzer or a roller pump and a column. And you'd be getting rid of all the organic protein-bound or not molecules that get across into dialysate. So it was a great idea. And in fact, in vitro, it worked beautifully. The in vitro binding toxin was very high, because the basic approach we used on all the other stuff was similar to that big green tank that's in the corner of your laboratory called mixed bed ion exchanger. So the deionizers that you use have a combination of a hydroxide-loaded anion exchanger and a hydrogen-loaded cation exchanger. Sodium chloride comes along, knocks them both off. They join and become water and disappear chemically. So it's a neat idea, but nobody had ever tried it as an oral sorbent. It worked beautifully in vitro, but it didn't work very well at all in animal trials. And we tried and tried to figure out why. What was about the gut. We couldn't replicate in the test in a beaker, and we really never did find it. But what we did find out was that if you gave them independently, just 12 hours apart, then we got the function you wanted. Well, so that's the whole course. Because of our discussions of and interest in PD, I didn't include the projects that I have done to include peritoneal dialysis. I do have a chapter in the book about it. And basically, all of our lives are complicated by this tank-off flow pattern. We may not realize it because right now there's no great alternative. But these two types of catheters have completely irregular and incomplete blood outflow of PD fluid. And even training patients and nurses is when the end of outflow is tedious. Cyclers have to have long drainer periods. And residual fluid means that you're losing chemical efficiency. So anyway, we've had a variety of projects designed not only for catheters, but other things, and I'll just go through them real quickly. There was the column disc catheter in the 1970s and 80s, successful. And it worked, but you had to place it surgically. And that was a big downside. You all know about the peritoneoscopic system I developed, just because I always envied the laparoscopists, pictures that surgeons could get. And we had to guess where our needles and catheters were. We did also, our lab made the model of how tank-off catheters worked and published it in a book by Alan Nissensen and others. And that became actually the main algorithm in the home choice machine, determining when it determined outflow. Surprised me, kind of scared me too. And then we did some wonderful animal studies in CFPD, but that wasn't necessarily a catheter. We did develop a catheter that I thought would have much improved drainage of the abdomen, which has little grooves on the surface and a T-shape. It did drain in patients and in the animal trials. It did drain absolutely completely, every last drop of PD fluid, but it wasn't any faster. And the reason was that T-shape was really inefficient to getting the fluid out the center part. So we ended up adapting this to a much more simple design of a tank-off type catheter like that. And in silicone and single body, you can straighten it out on a stylet, but it's a little larger than standard catheter. And this has incredibly increased in both rate of flow and completeness of drainage. And I hope it'll be in clinical trials this year. We've also done some work in bioimpedance. So anyway, what have we learned from all this? I hope something. And basically, in whatever career you choose, there's going to be frustration when the tools of the trade fail you and others. And you can resolve your frustration by finding out why it fails and thinking of an improved tool. The alternative is you can sit around the doctor's lounge and bitch about it, but that doesn't really get you very far. So you need to test your new idea, discuss it with others. If it's really sound, you got to build a prototype. And this is where so many physicians stumble. How can I build a prototype? I mean, I don't have a machine shop, et cetera. You collaborate. And it doesn't matter if it's the right size. It doesn't matter if it's crude. It doesn't matter if it's a chemical approximation. You learn a lot in building a prototype. So when you have results that's showing the promise, you find a company to collaborate with. You want to make sure the company really wants to change their product offerings. When you think about it, you're not going to have entry to a company that just loves its product and doesn't need to be any more successful. You got to find a company with a successful leading product where they got at least one manager or engineer or physician who says, this thing really sucks, and is really looking at the time for change to change things. And you have to stay close with it. So many physicians think, oh, great. I mean, I got a patent, I got FDA approval, we license it to a company. That's great. I'm going to the Bahamas. And it doesn't work that way, as you can see from the marketing failures. You have to realize if your real goal is market acceptance of the product, you're going to lose most of the time, actually. It's just like sports, if you could win 50-50, you're doing well. So what you do is move on to a new technology for that problem, or find a new application of the old technology that you've already been working on, but you don't quit. And you have to realize, I finally did, there's innumerable hurdles between your proven success of a device and widespread market, including steps all the way down to your own market not having it when other countries do. So many of them are out of your control, governmental regulations, patents, marketing, etc. But don't despair. Even if your idea or device doesn't make it to the market or stay there, you basically will stimulate others to prove its worth or improve it, or you'll invent something better and more successful. But basically, I think of each new device and drug that goes on the market as a thesis publication. When you track the technology backward, you'll find years, some decades of dedication of scientists and physicians to that product and idea. So you got to keep the faith. This is what you're made to do. So I thank you for your attention. This has been kind of a long and rambling thing, but I thought just knowing a little bit of the details of each of these stories would be helpful. And besides, now if you do get the book, just skim the middle chapters on each of the projects and read the introduction and the conclusions. So thank you.
So, thank you, Dr. Ash. That was, I mean, I've listened to many of your talks on different conferences, but learned a lot from this one. A couple of observations, which, out of, from an innovation pathway and, what made true success to the market and how it truly impacts our patients. I didn't realize zirconium was born out of a dialyzer or a dialysis machine, evolving into a product which thousands of patients consume now and keeps them off the hospital. So it's truly amazing to understand that piece. What on this journey, I did see that at some point there was some thought process behind a single needle cannulation. If you think about what could be revolutionary for vascular access now, one thing is catheters. We understand 50% of patients 80% of patients start dialysis with a catheter. And eventually they, evolve into a fistula. We also understand a substantial number of grafts, their half-life is anywhere between 4.5 to 7.5 years based upon how it's being used. Fistulas probably longer. A lot of this can change if we are able to somehow find that niche on getting it back to a single needle cannulation. Where did that fall off and why? And is there an opportunity for us to get back into that? What are your thoughts?
To your questions about the cannulation of
the single needle cannulation as opposed to dual needle cannulation. Think about it, cutting down almost 50% of every trauma we create to every axis.
Yeah. Oh yeah, I agree. And that I didn't really cover that, but that's been an amazement to me that we do not use ultrasound on all of our needle placements. Somehow we can work out the practicality of that. But I bought an ultrasound unit for our dialysis unit. I say our clinic did 30 some years ago. And if the nurses just couldn't cannulate a new fistula, if they had real problems, they would come over and ask me. But I would never go without the ultrasound. because it gave me an advantage that they didn't have. And quite frankly, in terms of the technique, the nurses are way better than most physicians, are way better than me at it. So it was just incredibly revealing. But in spite of the interest and the push for that by a number of people, Debbie Brower and other people, I think very, very few units have an ultrasound even suitable for needle placement or fistula evaluation, which is merely in their hands trying to answer the question of how big is it and how deep is it and where is it straight? What's the direction? So
but my question was more towards single needle cannulation, Dr. Ash.
Oh, yeah.
Cannulating 2 needles, if we have
Yes.
a technology that can take us in that direction. I saw that in your presentation. At one point there was, traction towards it on the early days, but where was it last and why?
Yeah. Well, you're asking why isn't single needle access being used more routinely? There really are limitations in it, which are pretty much mathematical. However, there's no doubt that sticking one needle in, and usually in the best location, is easier and much easier on the access. The real limitation is if you have a single needle and it's, say, 15 gauge, you can draw blood out at the usual rate, 350 mils a minute, but then you have to stop the outflow blood to return for time to return. So your overall blood treatment rate is going to be half of what you're treating today with flow through. Now, people have tried repeatedly, clear back in the 70s and 80s, to solve that by putting in a needle with 2 lumens. So at least you could have continuous treatment and maybe a little faster. But that's a problem that just doesn't work out when you realize how needles work and how they actually penetrate into a vessel is very is really sophisticated, starting with a tiny point, then there's a scoop and a widening part, and then a narrowing part as it goes back. So just that access to do it with a dual access device is difficult. Plus, if you put a second return line down the middle, it's going to have really high resistance because the concentric flow is great, but it does increase the surface area of contact tremendously for the blood. So I think that it goes back to how can we promote slower dialysis? And any way we can do that, it'll lead directly to single access, I believe. But if you go into a center and say, okay, I have patients successfully on 3 hours of dialysis, and now it's going to be 6. That's not going to fly. And so it's unfortunate. It's a topsy-turvy. But what we really decide is what schedule we want. And then we hammer dialysis procedures until they fit. There's no doubt also those patients on overnight dialysis or frequent dialysis at home are clearly healthier in all ways, and they've proven that. So it really ties together with home, which, by the way, is why the 2 hemo machines my company came out with were intrinsically single access. Because my real goal, my dream, was 8 hours of dialysis every other night. I think that's one of the most practical schedules you can have. One other advantage, by the way, of single access is it's an automatic protection for venous disconnect. In other words, even though it'll be messy, what'll happen is when the needle comes out, you're going to draw air into the inflow line, and it's going to shut the machine off. After a couple of cycles at most, you'll have a puddle of blood this big on the bed, but it won't be silently sucking blood. So anyway, that's the saga of single access, and the machinery is there and the needle is there, but really it's a matter of, can we tolerate cutting our blood flow in half by some increase in schedule somewhere, somehow?
Dr. Abraham, any thoughts? Dr. Abraham, you're muted.
Dr. Bala, actually, that was an interesting question you asked. I remember actually, bringing this out in one of the inform meetings, asking for some people. I was requesting many people who could talk on single needle dialysis uh, procedure, because I used to see this in many missions which I came across earlier days, but I never seen somebody using it. So why at all it went away? So the same thing what you asked now. But what I see now is the innovations have to come from advanced countries like US, and what I see in US is they are into more and more of shorter duration of dialysis. Actually, recently, right now, one of my patient has gone to US, and there their practice is to give 3 hours dialysis, maybe with 350 ml per minute flow. So I don't think somebody is going to work on single needle dialysis in US, because you are all interested to shorten the time and you know uh, in the dialysis room. So maybe places like ours, where sometimes this time may not mean much. I think we should really think about it and try to get around the problems and reuse this older thoughts about single needle dialysis.
And it's best said that the best treatment happens at home, right? If you're doing a longer treatment and if you have an option to use single cannulation at home, nothing can beat that. And in fact, Dr. Ash, Jeff and I, John Lampropoulos, Merit CEO, we're working on a prototype. I think I shared it with you 4 years ago on single needle with soft and hard edges. Truly, that will make a difference. But again, getting it across with all those different parameters you mentioned on flow and cycling will be relatively a challenge, especially if we can perfect the cycling mechanism of what's aspirated and what's given back across a split catheter. I mean, a split lumen single needle. That will make a difference. Dr. Georgie Abraham, you have any comments? You're muted though. It'll be helpful if you can unmute. So Dr. Abraham, we can see you're speaking, but you're muted. We can't hear you. Would you want to, if any one of you have a cell phone number, please text him and let him know, ask him to unmute himself.
Yeah, I'll call him.
So as we are trying to get Dr. Abraham back online, on the catheters, I know it's a necessary evil, but again, I shouldn't even use the word evil. It is necessary because that's where the reality hits the ground. And as much as we push for fistulas, which is great, we have more of our nephrology colleagues in India doing fistulas, are doing an amazing job. Making catheters even better, like making America better, right? Making catheters even better so that we don't, fuss and whine about we shouldn't be having long-term catheters. That's one thing. And of course, on the PD side, it's never breaken that bank of more than 20%, right? Despite all the advancements we have done. But unfortunately, there's not a lot of forth-looking advancements we have done in PD. So between catheter technology, I see you're putting a lot of effort there. I don't think it'll ever become a reality of 50% unless there is really a technological advancement on how those catheters work in terms of fluid extraction and fill-in and better clearance. It is not just the catheter, but also on what fluids we use. Any thoughts on that, Dr. Ash? And Dr. Abraham, again, if you can unmute yourself, would love to hear your thoughts on PD and how we can improve status quo.
Yes, just in answer to your question, I think that the most important first step in making progress is you really have to know the problem. You really have to live in the middle of it and understand why it doesn't work. And the reality is that the flow problems of the catheter are eventually limited by sheathing. So then sheathing is caused by irritation, touching, the very fact of touching the internal surface of a vein with a small catheter wipes out the endothelium in that area. So we need to figure out ways to work around that phenomenon. I do think actually something like this tributary access I talked about that goes into the side of a vessel like this. Yeah, like that sort of, without touching the inside, is could easily work. It certainly does work for both direction of flow in the superior vena cava of animals. But to get the arterial access, if you wanted that, which would be ideal, you could do the same thing. You just have to find a blood vessel branch that you could block off with a catheter, basically, to make it just stick in that far. And it'd be very much like an endoside graft that they use in kidney transplant, or something like that. And if we do that, then we're really paying attention to just how fragile that endothelium is. Whatever answer we get for long-term hydraulic function, going to have to address that problem. The infection part is also, we just have to admit and realize 80% of it is catheter hub contamination. So if you wanted to solve it, you need to put something in the catheter at the end of each treatment that will not only kill the free bacteria, but also the biofilm, eliminate it. And taurolidine, that new product, it will do that. But also citrate benzyl alcohol would do it. So, and you can buy that in the wrong concentrations right now from your compounding pharmacy. Everybody uses it. And in fact, legally, it's been on the market at 7% with 1% benzyl alcohol, and that's quite bactericidal. And it's in our purvey to order it. However, getting that done in a major LDO is not an easy step. So, so much for that. I do think there's an answer out there at the next stage for truly arteriovenous access.
Yep. And there are a few products that are, in the making, which, I can't disclose because
good.
I'm under.
Yeah, I'd be interested in your ideas. So
yeah, so that's evolving because everybody understands the reality of how much ever fistulas we want to create, but still there is a number which we all hit. What about preventive measures in PD catheters? And I'd love to learn from our colleagues here from India. What is the prevalence rate of PD there? I know at some point, at some conversations, Dr. Jairaj, in your lecture, you did mention about it, but I don't remember that. The prevalence rate of PD in India and what is the risk of infection? And Dr. Ash, then you can comment on, is there anything evolving to prevent peritonitis in that space? Dr. Jairaj, India, PD, and prevalence?
Yeah. Vineet? Vineet? Is Vineet there?
Yes, sir. Yes, sir.
Yeah. You respond.
So the PD prevalence is less than 10% of patients of RRT are on PD. So quite less in our country.
And what about peritonitis?
I'm sorry?
The rates of peritonitis?
So the problem is that because of lack of proper facilities and techniques, the exact rate of peritonitis as a country also we don't know. And from center to center, I think Georgie sir would agree. The rates may vary. I mean, there is a wide variation of rates. So very few good studies have shown proper rates, and generally, as it is a tropical country, the rates are generally high, and more than 50% of the peritonitis are actually culture negative peritonitis. So even that is also not well known.
Got it. I mean, all the more needs for something that could potentially be preventive. But that being said, the rates of peritonitis, which I've noticed in Indian patients here who I treat, is the least. So they do a phenomenal job taking care of the catheters. It's like, once you, tell them what you need to do, this is how you dot an I and cross a T, they will exactly do it the same way. And I don't see peritonitis in Indian patients in the United States. So they take extreme good care of the catheters. So that being said,
This-
again, unmet need, and the F, which is the father of frustration, peritonitis. So any new innovation or technology there to prevent peritonitis, Dr. Ash?
Is it because the family involvement which makes
Yes,
people-
extraordinary family involvement, plus also the diligence which our patients, maintain in, cleanliness in keeping the catheters clean. Dr. Ash, you're muted.
Yeah, and basically it's a great disappointment to me in some ways to say, yes, there's two available solutions that could diminish the rate of infection in IJ catheters, but there is no such thing for PD. And it doesn't really make a lot of sense. Realistically, if a catheter lock solution it should be used in someone like the taurolidine heparin combination that's on the market, in someone at high risk of IJ catheters infection, and the high risk PD patients should be the same. And it would be truthfully, it would be really simple to implement a clinical trial of a mixture of citrate and a catheter and a benzyl alcohol. You'd have to do a lot of animal tests and get it out there. But the infection principally in the whole hub isn't necessarily something you can treat once it's all together. It's in the process of screwing things together that bacteria are drug into the inside. So you would need, but if you had a compound that would wipe out the bacterial contamination of that catheter at the end of each exchange or a treatment, there's no doubt it would work. I hope we have one. On the other hand, there's plenty of things you just don't want to put in the peritoneum to do that. You have to be really careful not to mess up the delicate epithelium there either. But I do think there's tolerant solutions.
Any other thoughts, comments from the audience?
Dr. Yashwant is asking if there is any role for prophylactic antibiotics when there is a breach in sterility while performing CAPD.
No, that's always done. Whenever there is a difference, whenever there is a breach in the antisepsis, then we generally give a prophylactic antibiotics for a couple of days.
I agree.
I would also add that as a basic protocol, all patients are given a first dose of antibiotic before the procedure. And as sir said, whenever we feel that there is some contamination, most of us add a prophylactic antibiotic.
Dr. Stephen, we have just published, good evening. I think we have met many years ago. I don't know whether you remember me. We have just published our results of using 70% ethanol for treatment of CRBSI in tunnel catheters. We have found that the salvage rates are excellent. Almost 100% salvage we were able to do with 5 days of therapy, where we give ethanol lock for only 4 hours a day for 5 days, and then they are continuously being used in addition to systemic antibiotics.
Yeah, and the catheters don't clot.
No.
Yeah, and 70% ethanol.
Yeah,
not IPA. It's not isopropyl alcohol. It's ethanol.
No, ethanol.
Yeah, good. That's a much better choice.
Yeah.
Wow. Oh, good. And if you publish that,
I will send you my publication. Yeah.
Good. Thank you. I love it.
Sampath, have you looked at the propensity for central vein stenosis in these cases where ethanol was used for?
Bala, I have been using it in many of these patients. Repeated courses have been given over the last 5 years, none of them had developed central venous stenosis, and because these are all strictly staying inside, and then we remove it within 3 hours to 4 hours. So the leakage is very minimal.
And also the lock volumes are very minimal. I mean,
Yeah,
one thing we
very
want
minimal.
in a monitor that is for hemolysis, but again, the volume is so, that's I think where Dr. Ash was also getting to with the isopropyl alcohol. So it really shouldn't. And moreover, it's also a shorter duration of time, like 5 days. The cumulative toxicity is never going to be, accumulated. So great. That's congratulations on your publication. Any other questions, thoughts?
Somebody has asked your thoughts on probiotics in PD and anti-biofilm measures like DNASB to proteolytic enzyme flush to disrupt early biofilm.
And outside of I think Dr. Kumar, you were talking about the use of ethanol in IJ catheters.
Yeah.
Or PD. Yeah.
IJ.
So it probably would work in PD catheters too. I don't really
know the
know
peritoneal reaction, and I'm worried about the peritoneal reaction and sclerosis.
Right. Yeah, well, a few years in the lab and animal studies, and you'll figure that out.
Yeah.
But no, you're right. It has to be done cautiously.
Yeah.
And yes, but we aren't in possession of a truly biocompatible PD fluid either. So we're not starting at perfect zero. So anyway, no, I think there really should be a prophylactic that will prevent infections. But I would remind everybody that it really shouldn't be an antibiotic. I think you're going to eventually have resistance, either of bacteria or infectious disease experts. I don't know which is worse. But I think that this needs to be a mild antiseptic. And yeah, it should work.
There's a comment, Dr. Ash, there was a question on probiotics and proteolytic and proteolytic enzymes on. So thoughts though. It's interesting thought.
Yeah. I have to admit, I'm no expert in probiotics, although I'm continually amazed by what really goes on in the gut related to toxin generation and removal. No, I don't think I can be too much help, but there's certainly, there's something there in the bacterial mixtures that will help prolong the life of patients and possibly give them better health with kidney disease. And I think, if you look at the animals work, the veterinarians believe in the probiotics and also in the dietary treatment of kidney failure very effectively.
Actually, he's suggesting putting in probiotics in the fluid, PD fluid, isn't it? Is that you?
Yeah. Yeah, I have to beg ignorance of that. So I thought a probiotic was a it's a bacterial thing, but I think it's an interesting idea. If you're basically talking about a pre-drug, a prodrug, that might get activated in the body, that would be helpful.
Yeah. And considering the peritoneum is a sterile area, you get peritonitis only when there is those micro perforations happening in the gut. That's when you get the E. coli of the world. And but using a probiotic, which is basically bacilli in the peritoneum, again, it's something which we need to. Are we wanting to meaningfully introduce probiotics in the peritoneum? And it's a very interesting thought, because eventually, if there is going to be a micro leak from the gut leading to an E. coli introduction into the peritoneum, can the probiotic, do what it's supposed to do, so that it can prevent the E. coli or whatever organism can eventually create issues with the catheter? Very nice thought, really nice thought. And And on the proteolytics, in the peritoneal side, it might not help tremendously. And again, we have looked into what the biofilm, Dr. Ash is an expert there, how much can proteolytics help there? On the peritoneal side, it's not the physiology there of what actually creates catheter problems. On the hemo side, we don't know, it might, Dr. Ash, it might do something. And what is the impact when it gets systemically? Again, Dr. Ash, you're back on mute.
I think a major advance, by the way, in terms of the longevity and infection in PD would be the implementation of true nighttime cycler therapy. When that concept first came out in the late 80s, the idea was that you could hook up to a machine for 8 hours at night and get all the dialysis clearance you needed. And if you did that, then of course you'd be free of any fluid during the day. And in addition, I think the peritoneum would do a whole lot better at preventing these infections as well. The problem I see in that therapy is totally or mostly catheter related. Even with this tidal PD, you get 20 liters in and out of the belly. You're leaving a whole liter in the belly at the end just so the catheter will work right. So a catheter that actually would drain the abdomen in 5 minutes instead of 15 to 20, and drain it down to the last 50 mils instead of 500, would definitely help clearance so much that I think we could get back to the idea of just doing PD at night. In even the more prolonged or serious cases of ESRD, that'd make a big difference. We still have problems in PD with the long-term longevity of the therapy, and it's disappointing to me that the change in the PD membrane. Yeah. Yeah, it's disappointing to me that the with the loss of some mass transfer in the PD and the progression of intrinsic loss of renal function, we're still transferring a lot of patients back to hemo from PD at 3, 4, and 5 years. So I think we could reverse that also by true nighttime cycler and give the peritoneum a rest 16 hours a day.
Thanks, Dr. Ash. And one more question that I saw there, the role of surveillance blood cultures in patients, with the TDC, tunnel dialysis catheter for CRBSI.
I'm sorry, could you say that again?
The role of surveillance blood cultures in patients.
Oh, surveillance blood. Thank you. Yes. Probably. It makes sense on one hand theoretically, but the studies of biofilm in these catheters have shown that there's a rapid progression in maybe half the patients to establishment of some type of culturable biofilm if you scrape it all off. And yet that organism is rarely the same one as what causes the first peritonitis. So there's a disjunction there. I'm afraid you get what would be true false positives. And then again, a false positive if your criteria is, are they going to get peritonitis in the next few weeks? So truthfully, I think a better surveillance method would be to finally have an effective neutrophil assay device hooked to your cycler machine. And if you could pick up that first passage of that number of the granulocytes versus 50 per cubic millimeter, I think we'd treat it so much earlier with oral antibiotics even that it would help. So that's the surveillance I'd love to see. And they're getting closer, but again, getting the dialysis industry to realize the huge advantage for what to do when a patient calls and says my fluid's cloudy. It's been a little disappointing. And, the technology just based on turbidity or something simple, or maybe even total cell count isn't going to help you. You need to really know the granulocyte count.
Excellent.
And when do you foresee your new PD catheter design coming into practice? In what stage is it there? You said they are being used for chemotherapy, right?
I'm sorry, could you say that again?
No,
I had a little.
your newly designed catheter with
Yeah.
longitudinal grooves.
Yes. Oh, okay. Yes.
When do you
You're-
think? Yeah, you think it will come into use in the near future?
Oh, I certainly hope so by next year anyway. It takes a huge amount of time and resources to even bring a new catheter like that to production in a clinically usable form. And even to go into a clinical trial, you have to match all of the requirements for manufacturing, stability, residual testing, and all that sort of thing that you'd have for the final product. So that's progressing currently in Mexico. And the first use of that catheter won't really be in peritoneal dialysis. It'll actually be in another peritoneal therapy called HIPEC, which is hyperthermic intraperitoneal chemotherapy, done for people with advanced peritoneal carcinomatosis. And right now it's done by a complete surgical general anesthesia procedure where the belly's open, residual cancer is taken out. They put in vital catheters, treat them for several hours, and then open them back up and take all the catheters out. And I was approached to see if we could make this catheter a chronic catheter where that kind of treatment could be done once a month. So that'll be the first actual clinical use on a limited basis. But I would say certainly in 26, it's on its way.
We all look forward to it, Dr. Ash, because we need that.
Okay. Well, thank you.
Very nice. If there are no more questions, again, a phenomenal session. We learned a lot from you, and we continue to learn from you, Dr. Ash. So, let's all keep innovating, and resources are required for innovation, but it shouldn't be an impediment. So let's get inspired, and see ideas. It's just a seed idea that starts, and then it can evolve based upon how much, relatively obsessed you are and passionate you are to get it, close to the finish line. And then it's like a relay. When resources are on that bench, you pass it on to the next person and be part of the journey and get it to the finish line. So how we get there doesn't have to be 400 meters full cycle. It can be a 400 meters as well, but you're the same team. So I'm referring to all those athletics because I see how phenomenally well India performed in the Asian Athletics Championships. So again, thanks, Dr. Ash, for being there. Dr. Jayaraj, if you have any comments, closing comments.
Yeah, I think I should thank Bala, Jaji, and our guest speaker, Dr. Ash. I'm actually reminded of one event. Actually, Dr. Jaji brought Professor Robert Eady to one of our southern chapter meetings in India, actually Kanyakumari. This Professor Eady was a dermatologist. He was one of the longest person who was on dialysis going on to transplant. When he was a medical student, he got into renal failure. So going through his history, when he gave his talk, it was all from, his fistula was created by the innovator Brescia. Like that, each and every step in his CKD or renal replacement therapy life, and after that dialysis, it was all the history of nephrology was really brought out. That was from the patient perspective. And today, and we had Dr. Ash, who started his life so early, getting into nephrology and being associated with Dr. Willem Koff and things like that. So that reminded me how he was going through the history of development in nephrology, and as in and contributing to the innovations in nephrology practice. So we were so glad to have Dr. Ash today, and I'm happy I could introduce him to the Indian audience here. And once again, I thank all the people who joined the meeting today, and I think we'll call it a day.
Thank you all.
Great. Well,
Great
thank you for the invitation. I really enjoyed it. Thanks.
Thank you.
Thank you.
Thank you. Good night. Good night.